Hello, everyone. Welcome to this webinar as part of Shimadzu's 2021 Analytical Extravaganza. I'm Christopher Gillis, LCMS Product Manager at Shimadzu Scientific Instruments. I'll be your moderator today. We will hear from Dr. Rich Helm, and our topic is the role of LCMS in glycoscience research. Before we start, I want to share a couple of notes for our viewers. The webinar console has a variety of items to help enhance your experience and interaction with us. On your screen, the slides will appear on the left-hand side. Directly under the slides, you will see a resource list with links relevant to the material being presented today. You'll also see a section for questions and answers. Please submit your questions during the presentation, and we will answer them during the Q&A session. Just below the Q&A box are the moderator and speaker bios. You may expand the items here to learn more about us. On the right are the survey questions that you may fill out anytime during or after the presentation. Finally, at the bottom pane are the icons to bring up all these options in case they are minimized or hidden. That covers the webinar format, so let's get started. Again, if you're just joining us, I'm Christopher Gillis, your moderator. Today we'll be hearing from Dr. Rich Helm, and our topic is the role of LCMS in glycoscience research. Rich Helm received his BS degree from the SUNY College of Environmental Science and Forestry in 1982. His graduate work was performed at the University of Wisconsin and the USDA Forest Products Laboratory, and he received his PhD in 1987. After a postdoctoral position in natural products at Oregon State University, he returned to Madison in 1989 as a research associate at the USDA ARS Dairy Forage Research Center, where he helped develop tools for plant cell wall analysis. He was recruited to Virginia Tech in 1992, where he began to establish his own laboratory in wood chemistry. His research efforts shifted over time towards analytical biochemistry as applied to various biological systems, and he worked to establish the infrastructure for conducting mass spec-based metabolomics and proteomics work at Virginia Tech. He presently oversees core services for the Fralin Life Sciences Institute at Virginia Tech. He is also director of the materials facility for an NSF-funded materials innovation platform that focuses on glycomaterials. He's a member of ABRF, ACS, and ASBMB. Now, I'll hand it over to Dr. Helm.
Thanks for the introduction, Chris, and thank you all for attending the webinar today. As Chris said, and as my bio indicates, I'm an associate professor of biochemistry at Virginia Tech. My other main responsibility beyond my undergraduate teaching role is to manage the life science core services for the university. That said, I see myself more as a researcher educator than an administrator, and I've worked on various aspects of carbohydrates since I was a graduate student at the University of Wisconsin. In fact, my first publication was on the molecular weight distribution of commercial cornstarches back in, well, I guess I don't want to say when that was, except it was before there was such a time as email, and the term glycobiology had not yet been coined. I think that happened around 1988. I've broken this presentation up into three main sections. First, I'd like to spend some time placing glycoscience amongst the three main omics technologies to emphasize its special place, and also emphasize why advances in glycoscience are harder to come by than in other fields. I'll then very briefly talk about the operational limitations of core facilities at research institutions, and then spend most of my time going over some of the recent LCMS-based glycobiology projects that we've been involved with and are currently developing. These include a project related to the specific carbohydrate found in meat that's related to cancer, a suite of glycomaterials involved in Lyme arthritis, and a new, recently funded NSF Center on glycomaterials. Most of the work I'll be talking about will be generated using the Shimadzu LCMS 9030 high-res machine. Now I'll finish up with what the future looks like for LCMS and glycoscience, and then I'll take any questions you may have. Seems like every talk I give, I use this slide in various formats. I've modified it over the years depending upon the audience, but it works in some way, shape, or form for all life science disciplines because, at least to me, it is a pragmatic view on the data that we collect. If we move past the genetic code and into the first biochemical process, we hit transcription and the transcriptome. One can envision some rate of conversion of a gene to a transcript as well as some rate of degradation of that transcript. My first rule of biochemistry that I tell all students is a quote that I have at the top of that genomics box. A gene at rest stays at rest unless acted upon by proteins and/or metabolites. Now, some may take offense to that, but by and large, that's the case, as is the statement below the box. A transcript is what is and what may never be. Just because a transcript is present does not mean it will all be converted to a protein, and if it is, one generally has no idea the rate of conversion or the efficiency of that conversion. Nonetheless, if a transcript is translated into a protein, now we have the workhorses of the cells. That said, every protein has a life history and a half-life. That protein may be post-translationally modified, most proteins are, and there will be a turnover rate for both modified and unmodified proteins. And, in many cases, modified and unmodified proteins are in equilibrium with one another, an equilibrium that can shift depending upon multiple factors. The field of proteomics attempts to measure the relative levels of all proteins in a system, so-called global profiling, to aid in deciphering biological processes. There are issues with this from both the analytical and biological sides. First, there is really no such thing as global profiling because as soon as you utilize a method of extraction, you've exhibited bias. This can manifest itself at the protein abundance level and/or the number of proteins that are identified in any particular experiment. On the biological side, I tend to classify proteins in the category of what is and what may be doing something. Enzyme concentration does not directly correlate with enzyme activity, and many of us are biased by the fact that we think that more is more activity. There are way too many allosteric activators and inhibitors of any enzyme to logically conclude that a higher protein level means the protein is doing more. Now, at this point in time, you maybe think that I believe transcriptomics and proteomics are a waste of time, and nothing could be farther from the truth. They are important measures of biochemical processes. We just need to keep them in perspective. I will say, however, that attempting to correlate messenger RNA levels with protein levels in time-matched samples is a waste of time. There are just way too many variables to think about to have those two correlate. And my evidence for that is just look at how many papers come to that conclusion, and I've done that myself. That brings us to the metabolome. This is the one ohm that is closest to phenotype in any biological organism. Metabolites are predominantly formed and degraded by enzymes, and in most cases, their levels do indicate cellular state. That said, it is one of the hardest ohms to measure, as there is huge diversity in chemical features. If one wanted a global metabolome by LCMS, one would need at least 3 different types of chromatography in both ionization modes, and even if you had that, you would not be able to identify them all because the databases are not complete. We have a decent understanding of the number of human genes, a reasonable estimate of the number of proteins, but just back of the napkin estimates for the number of metabolites, and if they are from human metabolism or the metabolism of organisms that reside in and on us. The reason I'm spending so much time on this is to place glycoscience in the context of these ohms. Genomics generates databases using advanced technology at relatively low cost. Proteomics utilizes these databases using relatively advanced technology, and I say relatively because I'm continually amazed by the advances in mass spectrometer technology. Once you think it has matured, out comes something else that is faster and more sensitive and at higher resolution. Metabolomics has no answer key in the back of the book. You end up generating features or retention time and mass-to-charge ratio pairs that must be used for manual or automated identification. And even if you do get an ID, it's sometimes too far away from a gene or a protein to make a clean biological statement. It's just a potential biomarker, and why many times funding agency review panels call metabolomics experiment a fishing expedition. These sorts of studies must often be done before submitting a proposal. All of the omes listed here have carbohydrates involved. The backbone of DNA and RNA, carbohydrates. A large percentage of all proteins are post-translationally modified by carbohydrates or glycans. And many metabolites are carbohydrate-based. Carbohydrates are the most abundant molecules on the planet, yet, by and large, it is a somewhat neglected or understudied field. Well, why is that? Well, basically because it's just hard work. Structures are similar, yet different, and good tools for their study are just hard to come by. The field is then left to those with strong desires to work in a difficult space, and hence there are way more folks that study other things besides the glycome. Protein databases or proteomic studies are just the starting point for glycoproteomics. There's no answer key there. And while the literature says it's easy, I beg to differ for complex mixtures of N and O glycans. And if you have a glycan or a polysaccharide that needs a structure determined, you're going to need a suite of skills that not many laboratories maintain. Alpha versus beta, D versus L. It's a stereochemical and isobaric nightmare. The above said, glycoscience is not, and I repeat, not lost in the wilderness. Computational tools and databases are becoming more and more mature, so much so that being a glycoscientist is becoming mainstream, but still hard work. There will be some streamlining and standardization of resources. That is what all the other omes did and continue to do, but we are a lot farther along than we were, say, 5 years ago. The next few slides are an attempt to provide a high-level view of the LC and MS portions of the tool for so-called best practices with respect to any glycoscience project, with the exception of monosaccharide analyses. There are much better tools than LCMS for separating and quantifying monosaccharides. If you have a glycan in solution that needs to be analyzed, the first thing to consider is the reducing end and whether or not it is necessary to reduce it to an alditol. This is a relatively straightforward process that most would consider quantitative. Reducing end groups undergo the process of ring opening and closing, interconverting between the predominant forms in an aqueous solution. This can lead to poor peak shape or at times a separation of the main forms. And one of the examples I will provide today involves an example of the ability to separate anomers. Reduction options include borohydride or borodeuteride, or a two-step process of reductive amination. Most fields of glycomaterial analysis have opted for reductive amination, as the process puts a tag on the molecule that will aid in ionization, aid in chromatographic resolution, as well as provide a spectroscopic feature that can be followed if purification is needed. Chromatography media can be classified into three separate groups: the standard reversed phase, such as C18, hydrophilic interaction, and porous graphitized carbon. C18 suffers from poor retention, and separation of related structures can be challenging for carbohydrates. The mechanism of Hylic involves a shell of hydration around the solid support. Running gradient separations in Hylic mode tends to need longer times of re-equilibration to reset the initial conditions, and peak widths tend to be much wider than what we've become used to for most chromatographic separations. Porous graphitized carbon is basically the modern-day activated carbon, which was the solid support serving as the go-to material to chromatograph carbohydrates a generation ago. While this system offers the best separations, it can suffer from variable retention times and actual loss of resolution. Various maintenance protocols have been developed to deal with the issue, and most are application-specific. Mixing and matching columns can offer increased separation capabilities. These combined techniques tend to use the first dimension in fraction collection mode, with each of these fractions then analyzed in the second dimension. While this can improve separations, it also dramatically increases the complexity of the separation and the downstream data analysis. When looking for a mass spectrometer to hook up to an LC, cost is always a factor. However, if one is serious about the analysis of glycomaterials, high resolution is mandatory, with my ballpark minimum of around 30,000. Clearly, higher resolution is better, and one will need to review the cost-benefit of moving higher up the scale of resolution and the subsequent scale and price. There are some other features and methods that also must be considered for glycoscience work. Improving the separation of similar species can be done in the mass spectrometer using ion mobility. In ion mobility, the molecules are separated based upon their volume in the gas phase. This often allows isobaric species to be separated. The output of an ion mobility experiment, or the collisional cross-section, is a physical property of any molecular species. And tables of CCS values, or collisional cross-section values, are available online for many glycans and oligosaccharides. Since many glycoproteins are at the surface of cells, it is also possible to release glycans from intact cells using mass spectrometry-based imaging. In this method of analysis, there's no LC component. Cells are plated on a surface, coated with a MALDI matrix, and analyzed by a MALDI TOF mass spectrometer. Brooker recently released a MALDI-2 device where a second laser is used to help charged species released from the plate after the first pulse. And this can increase glycan sensitivities by several orders of magnitude. Finally, we're left with the best features associated with ion fragmentation. Glycans are notoriously unstable in the gas phase and will suffer from in-source decay. By and large, that cannot be avoided, only mitigated. There are a wide variety of methods to fragment glycans, and most are vendor dependent, and I'm not going to comment on each one. Just suffice to say that all can provide consistent and reproducible fragments for analysis. This is a very active research domain, and I would not be surprised to see more advances here in the near future. Now, I don't know all of the roles you play at your respective locations, but I'd like to point out some inherent limitations at a university setting. Most universities have adopted course services, and Virginia Tech is one of those. And on the mass spec side, we're responsible for metabolomic, proteomic, and quantitative LCMS analyses for the entire university. The queue can get quite long at times, and we're required to bill for everything based upon rates established by the university. Glycoscience, as I had indicated earlier, is hard work, which really means a fair bit of LCMS time. As method development and data analysis can be a significant chunk of time, entering into a glycoscience project is sort of a gamble because it may not work. And customers do not like to be billed for things that do not work. In order for LCMS and glycoscience to move forward, especially in core facilities such as mine, we need direct collaborations between those in the trenches of glycobiology and glycoscience and those that operate LCMS facilities. These conversations need to focus in on what does the customer really want and what the resources can deliver and how much time, effort, and money that's going to cost. We have several different options in the lab for LCMS, but only two that are targeted, both of which are Shimadzu. We tend to use the triple quad for targeted analyses of known materials as long as labeled standards and established methods are available. The LCMS 9030 is a high-resolution instrument that can be used in multiple modes for quantitative work. We tend to use this instrument for experiments where there is no real method in place and we need to start from scratch. This is because we find the high-res feature useful for working through potential ions that can lead to erroneous quantitative analysis results. As the price point of high-res mass spec machines continue to go down, you'll find more and more labs migrating to the high-res platform over the triple quad platform just because of the increased information one obtains with a high-res instrument. And while generally high-res based quantification methods have a lower limit of quantification relative to a triple quad, this is not always the case. What is for certain is that once a lab sees high-res mass spec data, it's really hard going back. The first project I'd like to describe is a fully targeted analysis. Back in 2015, there was a paper that was published in the Proceedings of the National Academy of Sciences indicating that, quote, a red meat glycan promotes inflammation and cancer progression. Obviously, this is one of those papers that gets a fair bit of publicity. Meat causes cancer type of thing. Such research results put the industries associated with meat production in a bad light. Or in the very least, one needs to determine if indeed a red meat glycan promotes inflammation and cancer progression. We were asked by the beef industry to look into this in collaboration with a mass spec lab at Colorado State University. Our approach at Virginia Tech involved taking advantage of the high-res mass spec feature of the LCMS 9030. Here's an overview of the monosaccharides present in most mammalian systems. The compounds that were the target of this study were the two neurominic acids indicated in the box, oftentimes referred to as NEU5GC and NEU5AC. As one can see, the structures are very similar. The 5-glycolyl neurominic acid is the hydroxylated version of N-acetyl neurominic acid. These monosaccharides can be found free in tissues or, more commonly, present on the non-reducing terminal ends of glycoproteins. The project involved measuring both free and bound forms of these sugars. As I've already said, sialic acids are located at the terminal ends of the oligosaccharides attached to glycoproteins or free in tissues. The 5-glycolyl is synthesized from the 5-acetyl neurominic acid by a hydroxylase. Interestingly, humans have lost the ability to make the 5-glycolyl, making only the standard N-acetyl neurominic acid. But almost all other animals synthesize the 5-glycolyl sialic acid. Therefore, meats contain this molecule, as do therapeutic biologics, since they're mostly synthesized in non-human cell lines. Most humans therefore consume materials that contain the 5-glycolyl neurominic acid. That's a fact. If the 5-glycolyl is available, it can be activated and incorporated into human glycoproteins, and cancer cells have a higher level of the 5-glycolyl relative to most human cells. These are both known facts. Now, one could argue that the 5-glycolyl incorporation into cancer cells is just due to the fact that cancer cells are fast-growing and incorporate whatever is available. But since this is a foreign sugar typically found at the non-reducing termini of glycoproteins, they could elicit an immune response, which can lead directly to inflammation. Therefore, determining levels of the 5-glycolyl sialic acid in beef is important to that industry. Cows that were reared for a feeding study at Virginia Tech were used to assess sialic acid contents in several different tissues. There were two different cuts of meat. One was a ribeye cut, and one was one that would be more amenable to, say, ground beef. Also, we included liver tissue as well as the tallow or the fat from a particular portion area from the cow. The point of this was then to provide concentrations in these meat samples as well as in available standards, so we included NIST standards for comparative and standardization purposes. So NIST has a beef liver standard available as well as a meat homogenate sample, and we included those in this particular study. As a land-grant institution, Virginia Tech has a slaughterhouse on campus, and we used that particular slaughterhouse to collect samples from 16 different cows and these 5 different tissue types. Now, going into this, we knew that animal variability was going to be more of a problem than technical variability, so that is why we used so many different animals in this study. These samples were then ground in a commercial blender using liquid nitrogen and freeze-dried to a constant weight. A known amount of this material was then pulverized in a tissuelyzer 2 using stainless steel beads and water containing the internal standard that was available, the C13 version of the 5-acetylated neurominic acid. There is no standard available for the glycolyl sialic acid. The sample was collected by centrifugation and passed through an SBE column, taking the flow through. This was only done to eliminate the lipids that are present that we could easily eliminate by passing it through the SBE. At this point, that sample was ready for analysis, and that would involve the free sialic acids. The remaining material that was left over after the extraction of the free sample, we're going to classify as the bound sample, and that was washed to remove all liquids that were present, and then acid hydrolyzed with an internal standard added in, the same one that we used before, and we used 100 millimolar TFA, or trifluoroacetic acid, for that hydrolysis. Now, the acid hydrolysis conditions were optimized for each tissue type, and it depended upon whether it went for 60 or 90 minutes. But nonetheless, acid hydrolysis occurred. We released the monosaccharides, and we collected the bound sialic acid sample by just centrifugation. We took that back down to dryness to get rid of the TFA. We then did a SBE cleanup, which is basically the same as before, just passing it directly through to eliminate any material that would bind to a C18 column, and then that sample was ready for analysis. Now, I realize there's a lot of information on this slide, but basically I wanted to bring home the point that we were using high-res mass spec methods for quantification, and we felt like we needed several events to monitor the separation in general, as well as events to quantify the targets. So our first event was basically just to scan the mass range, so we had a good understanding for all of the species that were present. Events 2 through 4 were our so-called quantifiers for each of our species, and our events 5 through 7 were MS-MS experiments that essentially served as qualifiers. While we took the areas under the curves of events 2 through 4 for quantifying the particular species present, the MS2 spectra that were obtained in events 5 through 7 were reviewed manually to ensure that the quantifier events only collected data on our species of interest. This slide here has a picture of a typical result from the analyses that we did. We kept our run times as short as possible while still obtaining a near baseline resolution with a total run time of 15 minutes. There's a few things I'd like to point out here. There's no reason to collect data that you don't need, nor is there a reason to send sample from data that you don't need to the mass spec. We stopped our data collection after 5 minutes and sent sample to waste between 5 and 15 minutes. Though our column choice was basically limited to what we had on hand, the phenylhexyl column is actually quite reasonably effective at separating carboxylic acids. As with most columns, I tend not to go to 100% aqueous or 100% organic. Note, however, that we had to go to 1% organic to retain the sialic acids. And also note the use of methanol. My lab is attempting to switch to methanol for most applications just due to the sheer cost of acetonitrile. And you can see that when we collect the selected ion monitoring data, the runs are reproducible with real samples. And also note that you can clearly see the alpha and beta anomers for all compounds. And while this looks extremely clean on a per sample basis, the data is quite a bit noisier, and this is due to the animal-to-animal variation. Nonetheless, we can glean some information from the data that we obtained. Using the high-res mass spec approach, the 5-glycollyl is highest in liver tissue, with relatively little in fatty tissue. There is quite a bit more of the 5-glycollyl in the bound form, and the ratio of the 5-acetyl to the 5-glycollyl is quite variable between sample types. And when it comes down to a final item that the beef industry would be interested in, a quarter pound of ribeye would give you approximately 10 milligrams of the 5-glycollyl. The limit of detection in our system was 50 nanomolar, which is about 16 milligrams per liter. In summary here, we used the high-res mass spec technology in place of a traditional triple quad method for an individual carbohydrate with a clear mass and retention time. Overall, the process of moving through the protocol from start to finish was much easier than working through a triple quad approach. And a cursory look at the high-res mass spec to the triple quad in comparison indicated, at least for this case, the high-res method was more sensitive. My next example is a project that needed to be started from scratch. Lyme disease is a widespread epidemic in the United States with approximately 450,000 people diagnosed annually since 2010. If you know folks in the Northeast or Upper Midwest, you know someone that has been infected. Lyme disease gets its name from the location it was originally detected, which is Lyme, Connecticut. The causative agent of Lyme disease is the bacterium called Borrelia burgdorferi. A few years back, our Department of Biochemistry hired a new assistant professor named Brandon Jutras. His postdoctoral work at Yale involved a material called peptidoglycan from Borrelia burgdorferi. And what you can see here from a figure from their PNAS paper from 2019 is the ankle joint of a mouse that was injected with a control and purified peptidoglycan from Borrelia burgdorferi. One day after injection, there is a clear inflammatory response. There are tools available to assess the level of arthritis in mice, and that data is reported to on the graphs to the right. Borrelia peptidoglycan results in higher scores for arthritis and histopathological response. Since peptidoglycan was being added in, a molecular component from Borrelia was causing this. So the question then became, what is in this peptidoglycan? Microbes use peptidoglycan like we use spandex or tights. It helps keep us all together. Different kinds of bacteria use peptidoglycan in different ways. Gram-negative bacteria, or diderms, place it in between an inner and outer membrane, whereas other bacterial types keep it outside their single membranes. As a spirochete, Borrelia has both an inner and an outer membrane, and so the peptidoglycan resides in between those two zones. Note that spirochetes like Borrelia are long, skinny, and tend to adopt a somewhat spiral shape. They're one of the fastest moving of all bacteria, which to some degree is related to their physical shape. What is peptidoglycan? Well, it contains both peptide and glycan components. A disaccharide repeat of N-acetylglucosamine, beta-1,4 linked to N-acetylmuramic acid, is considered dogma, and there has never been an exception to this structural repeat. Peptidoglycan chains terminate with a 1,6-anhydromuramic acid, which essentially blocks the reducing end. The muramic acid is also linked to a stem peptide, the structure of which is species dependent. This stem peptide can cross-link across individual glycan strands to make the mesh-like network that's known as peptidoglycan. The 2019 paper where peptidoglycan was linked to Lyme arthritis also had a figure where the main components of the Borrelia peptidoglycan were listed based upon their standard method of analysis. This is a LCUV-based method with a phosphate buffer, and this has been standard for at least 20 years. The fragments that are listed are called muropeptides, and they are obtained by an enzymatic treatment of the isolated peptidoglycan. These muropeptides are then reduced with borohydride, and a suite of structures are formed. Let's look at the structures that are below the UV trace to get a feel for the structures that are present. Structure 1 shows a muramic acid moiety in reduced form and its stem peptide. The stem peptide is comprised of alanine, glutamate, ornithine, and glycine. Now, the stereochemistry is listed for each of those. That has never been proven with the samples that we looked at, but nonetheless, other studies have looked at it and have concluded that those are the isomers that are present. If one were to review the structures in whole, one can all see that they have glycan and stem peptide components, and some, like structures 6, 8, and 9, show muropeptides that have the cross-link between adjacent peptidoglycan polymers. Up to now, all known structures of peptidoglycan contained a repeat of glucnac and murnac linked beta-1,4. But look at structure 3 in the box. It is a muropeptide from the so-called reducing end of the peptidoglycan, as it has an anhydromurnac, as indicated by the A and H designation, but more importantly, there's an unknown hexnac shown, which indicates a non-canonical structure. Normal PG would have another murnac instead of the hexnac. Also evident and associated with PGNs are the isobaric structures circled in red. Those isobaric structures have very different retention times, yet similar structures. We sought to investigate these odd structures while also generating a database for Borrelia muropeptides from several different Borrelia species. Here I provide an overview of how the samples were prepared for analysis and an overview of the data interrogation. Peptidoglycan is very insoluble in most solvents and can easily be obtained as a solid by exposure to boiling SDS solution. This sample can be treated with commercial enzymes to release the muropeptides. In our case, we used an enzyme that cleaves glycosidic bonds between murnac and glucnac residues. This leaves the non-terminal murnac residues, or the reducing end, that can be reduced to an alditol with either borohydride or borodeuteride. This reduction simplifies the chromatography. The LC and MS details are also shown and relied upon the Shimadzu Nexera XB3 UPLC interfaced with the LCMS 9030. We used a standard reversed phase UPLC column and a gradient from water to methanol under acidic conditions. High-res data was collected in multiple ways, either in data-independent or data-dependent modes. The Shimadzu data was easily converted to data formats amenable to both open source and commercial software tools. Since we have a core facility that has a training mission and we do not want to spend all our time analyzing data, we typically use MS-Dial and MetaboAnalyst for data processing and analysis. This allows for investigators to review data at their own desktop so they do not need to tie up facility computers for their own data analysis. The graphical user interface keeps folks new to mass spec data engaged, as it's real easy to lose them at this stage of the work. It is relatively straightforward to move from raw data to feature identification and comparison of sample types. In fact, the data shown here can be generated in less than a day after data acquisition is complete. MS-Dial can quickly visually represent feature differences, and here I have shown it for 3 different Borrelia species. MetaboAnalyst can generate overall views of the data, here showing each species have different overall profiles. Bionic, which is commercial software, can help interpret the MS data, leading to an initial structure. Interestingly, all of the species present contain the novel muropeptide with the unknown hexnac. An example of the LCMS separation of the muropeptides is shown here. This led to 27 different features for identification. We then went about collecting key data for these muropeptides, building an MS1 and MS2 database. Here we have the tick and associated extractant ion chromatograms for several of the unknowns. The MS2 database contained key fragment ions, as well as data for whether or not the muropeptides were internal to the peptidoglycan or at the reducing end where they would be in an anhydro form, using reduction with borodeuteride and borohydride in separate experiments for mass shift confirmation. With this in hand, we could then address the unknowns of interest, the first being the non-canonical hexnac containing species. The evidence we obtained by a mix of classical carbohydrate chemistry and LCMS is shown here. Acid hydrolysis of a muropeptide mix only resulted in detection of murinac and glucanac. Feeding trials with mannac and galnac and their subsequent analyses, either by acid hydrolysis or LCMS, only revealed muropeptides with glucanac and murinac. Feeding trials with glucanac or mannac labeled with C13 at the C1 or anomeric position found the same muropeptide pools, with the unknown having a mass increase of 3 Daltons, implying that there were 3 carbohydrates present in the structure. From this work, we concluded that the unknown hexanac was glucanac, and Borrelia has a novel peptidoglycan structure at the termini of its peptidoglycan chains. The isomer work is still unfolding, and we will use a combination of LCMS with ion mobility and NMR of labeled peptidoglycan to decipher that. So in summary, LCMS played a vital role in starting from a biological system and developing tools to assess structure of a material that may play a direct role in a disease state. One of the limitations in glycoscience research is the availability of resources at both the sample level and the bioinformatic level. Virginia Tech and the University of Georgia were recently awarded a 5-year grant to establish a national user facility for glycoscience. The main missions of the facility are to make available tools for glycan synthesis, glycomaterial structure determination, computer modeling of glycomaterials, and molecular interaction measurements involving glycomaterials. The long-term goal of the facility is to develop new glycomaterials that can benefit society. The Materials Hub is located at Virginia Tech, whereas the University of Georgia houses both analytical and modeling resources. Of central need to the glycoscience community is a way to synthesize glycans in automated fashion. While the automated word here is used in parentheses because the work is challenging, it is possible to create a suite of materials for use in both glycobiology and materials of use for modeling and/or use in society, so-called glycomimetics. As an example of the nature of the problem, this slide shows state-of-the-art for nucleic acid, peptide, and oligosaccharide synthesis. The cost of putting 2 nucleic acids together is minimal, approximately 10 cents US per base pair. A peptide bond can be synthesized for about $2. There is no present price point for a glycosidic bond, as we have multiple sugars, multiple linkage points, and 2 ways to orient the glycosidic bond. We have recently purchased 2 automated glycan synthesizers from GlycoUniverse as part of this GlycoMIP National User Facility. GlycoUniverse is a company based in Germany that has commercialized the work of Peter Seeberger. This tool can be used to generate glycan arrays, standards for modeling, and database generation for both NMR and mass spec, labeled carbohydrates for glycobiology work. One can mix chemical and enzymatic syntheses for more complex structures, tools for enzyme assays, and new materials with particular structure-function relationships. While this slide is busy and shows all of the different chemical features associated with automated glycan synthesis, the process is quite simple and is based on the fact that the oligosaccharide synthetic process is repetitive by nature. A building block can be attached to a resin and a series of deprotections and glycosylation reactions can be used in sequence to assemble a protected oligosaccharide. This protected oligosaccharide can then be removed from the solid support, purified by preparative LC, and deprotected, followed by a final purification to release a target compound with LCMS and NMR used to confirm structure. As exemplified in the slide here, synthetic oligosaccharides can self-assemble with different morphologies depending upon length, substitution, and assembly method. I show this only to bring up the fact that we have yet to scratch the surface on what biomimetic glycomaterials can do for advancing the field of glycoscience. As an example of where we can go with glycomaterials, I provide a few slides on a direct application of interest to me. As you are all aware, the cold chain is an issue for biologics. For a number of years, my laboratory was involved in understanding desiccation tolerance in microbial systems. You have all seen desiccation-tolerant species, most notably on the roofs of asphalt shingle homes. These microbes withstand complete desiccation and direct exposure to UV light by encasing themselves in an extracellular polysaccharide. All they need is some water to come back to life, gaining nutrients and minerals from the place of their growth. In the past, we have solved the structure of one of those extracellular polysaccharides and utilized the approach to preserve cells as well as prepare biosensors that can be stored for long periods of time. It is projects such as these that can advance glycomaterials beyond its present state, which is one of the main objectives of the GlycoMIP facility. Some final thoughts and points. Glycomics has lagged behind other omics fields, but it is catching up. Advances in our understanding of glycobiology have relied upon LCMS technologies and downstream bioinformatics tools and will continue to do so. The two examples I presented today, one on the analysis of meat samples and another on analysis of materials from a Borrelia burgdorferi, or the causative agent of Lyme's disease, shows how widespread glycoscience research is. The field of glycoscience is expanding at a rapid rate. There are more and more investigators joining into activities related to glycoscience. And as technologies develop further, we are going to see glycobiology and material science merge into new lines of research that will lead to new knowledge and products that benefit society. It's actually a great time to be a glycoscientist. With that, I'd like to close with a list of sponsors of the work presented here, as well as introduce the names of my team. My team at the Mass Spectrometry Incubator is small, but productive. Sherry Hildreth works on small molecules, and Keith Frey works on proteins. But unfortunately, I didn't have time to discuss our glycoproteomics work. The Beef Chekhov Foundation provided funds for the sialic acid work. The Cohen Foundations provided funds for the Lyme disease work. And the laboratory of Brandon Jutras and his graduate student Tanner DeHart has provided the samples to my laboratory for analysis. The lead PI for the GlycoMIP project is Maren Roman of our Department of Sustainable Biomaterials, and the leads at the University of Georgia are affiliated with the Complex Carbohydrate Research Center, also known as the CCRC. And finally, I would like to thank Shimadzu for introducing us to their LCMS 9030 and the opportunity to speak to you today. Thank you.
Let's thank Dr. Helm for an outstanding presentation. Again, under your resource list, you can find more information about our analytical solutions. If you have not had a chance, please fill out the survey questions on the right-hand side of your screen to provide feedback and request additional information. We also encourage you and your colleagues to attend our upcoming webinars, which encompass a wide variety of application areas. The registration link is located on the bottom of this slide. Thank you all for attending. At this point, we'll go to our Q&A session for Dr. Helm. Our first question is, Dr. Helm, do you see glycoscience as the future to look for cancer biomarkers since proteomics research has fallen short of early 2000s expectations, identified far less proteins than thought?
Well, I think it's going to have to be a mix. There are certain problems that still need to be solved with respect to actually getting useful information from a glycoproteomics experiment. There are so many variations on one protein for a glycan that it's difficult to say what's the most important or how do we rank them with respect to their structure and then the wide diversity that's there. And we can see that even just the COVID particle. If you were to look at the number of glycans that are on the COVID particle, and those glycans are put on by human enzymes, they're vast. There's hundreds of different variations on a theme, and each of those are going to have a different immune response, and each of those are going to affect each individual differently. So it will have to be a mix, and somehow we got to have a better way to rapidly measure glycan profiles on all types of cells.
Thank you. In fact, I'll add my own follow-up question to that. When you were discussing metabolomics, you mentioned that there were 3 types of chromatography needed for a full analysis of metabolomics. Could you elucidate on that briefly?
Well, basically, I sort of bucket the metabolites with respect to their polarity. Right? We have the things that are highly polar, the things that I would call semipolar, and then the lipids, or the components of predominantly cell membranes. So that those polar materials, they're best looked at by the PGC or the Hillock type methods because they're not retained on a reversed phase column. Reverse phase columns are fairly decent at semipolar compounds, or things that at least have some adherence to a reversed phase solid support. And then the lipids, they don't work too well on a reversed phase, and there's standard conditions that need to be used for those to get them separated out. So there's standard, relatively harsh organic solvents that are needed to fractionate them in a reproducible fashion. So you basically need, if you want to characterize all the metabolites, you really need 3 different modes of chromatography to capture the polarity that's involved with metabolites. And then you need both ionization modes. So basically you need 6 data sets if you want to get a quote-unquote global profile.
Thank you. And do you tend to avoid ion pair as much as possible?
Yes. I just find that in the end I would prefer not to have anything in there that's going to contaminate my instrument.
Okay, thank you.
So quaternary amines, I don't allow quaternary amines on my instrument.
Okay. I would like to say neither do I, but sometimes sometimes we can't avoid it. We have some other questions coming in here. How popular is the mixed polarity run in glycomics?
There are specialty labs that do a very good job at that. It's not something that's done routinely. The same thing can be, proteomics is the same way. We can take fractions and increase the coverage. I have found that it's easier to do a shotgun approach, if you will, to first find differences, because if the differences are down in the weeds, I don't know how useful that information would be. I would rather start at the top, find differences from a single separation, and then figure out the best chromatography for that particular metabolite. I have not seen any core labs do this this combined or orthogonal fractionation approach just because of the complexity associated with it.
All right, thank you very much. Here's another question. Can you describe the process of adding a compound to your MS-MS library? what qualifications did you require?
Yeah, so the way we limited it was we basically did peak integrations based upon the total ion chromatograms, and again, we didn't have standards to get a mg per mil, but we could at least get a relative area. And then we decided anything greater than 5% we would attempt to put in the database. Anything below that we left alone. So that was just a broad cut to limit the numbers that are there, because when you get farther down into the weeds, there's many, many more.
All right, thank you. Here's another question. I apologize if you addressed this in the presentation. Do you have a sense of the diversity of glycosylations in a given disease state? Could there be 2 variations, or 10s, or hundreds?
I have seen reports of one particular protein in a human cell line having 73 different modifications that can occur over time. And so the variability associated with one protein and the suite of glycans that can be present are enormous. And then this is an extremely difficult area to work in, because then their levels are low, and then you don't, if you were to cleave them off, you don't know where they come from. And if you were to produce a glycopeptide, you can get that information, but it's oftentimes difficult to place that particular glycan moiety on the right serine or threonine and decide whether or not it's N or O linked. We have a decent way to cleave off N glycans, but O glycans are much more of a problem.
Okay, thank you. Just adds to the complexity of the work.
Yes.
You mentioned, and I thought this was interesting. You mentioned that customers do not like to be billed for projects that don't work. What questions do you ask to qualify potential projects before you take them on?
Yeah, so my general rule here is to under-promise and over-deliver. That's the general operation, how I tend to operate. And so in essence, what we try to do is at least set expectations so they understand that there's certain things that we are pretty confident and can be done, and certain things that might not be done, and then we'll set a estimated cost for that based upon those issues, and we have to agree upon that before we move forward. I've never had an individual not pay, but I've also had individuals that have gone away somewhat frustrated.
Does that mean that you need to see, for example, a chromatographic method developed before you'll take on the project, or do you just assess your own lab's ability to handle those aspects?
Right. So then basically I try to set it up in phases. Phase 1 is what we call the project setup fee. Can we at least get to point A? Right. Then we need to make a decision. If we get to point A, can we go to point B? And so I try to have enough communication with the folks on campus to make sure that we're all on board with respect to where we sit, and then do we need to make a decision to go forward, or do we stop right here? Sometimes there are situations where I say we just can't. I just don't feel comfortable doing that type of an experiment.
I like the way you broke that into milestones. How do you avoid project creep?
Oh, that happens all the time. The client will say, I have 6 samples to analyze and 12 come in the door. Right, that happens all the time.
Right.
And so it's a matter of just realizing that that's going to be a factor. And then if you do things, we charge based upon hours on the mass spec, and then that sort of quantifies it. Right. If they had 6 samples, they said, and they deliver 12, that's twice as much time. Right. And they need to understand that before we go.
All right. Well, thank you. Well, it looks like we're running out of time here. Thanks everyone for the excellent questions, and Dr. Helm, thank you again for an outstanding presentation. If there's questions that we haven't gotten to, we'll reach out individually to each of you whose questions were not able to be answered today. And so once again, thank you all for attending and participating. We'll send you an email with a link to view a recorded presentation, a version of this presentation that you can watch anytime. And again, we encourage you and your colleagues to attend our upcoming webinars, and the registration link is located at the bottom of the slide. Thank you for attending, and have a great day.
All right, goodbye.
Thank you.